SensUs competition of 2025. It is fantastic to see all of you here, whether you're joining us in person or online. Before we begin, I would like to take a moment to thank all of you, our guests, partners, and of course, the participating teams. Your passion, dedication, and creativity are what makes SensUs truly special. We have an exciting program lined up for you today, starting from the general opening, followed by a presentation from our sponsor PalmSense on how to accelerate your journey from the lab to reality. This will be followed by then the panel talk, then by a presentation from the company LinkSense, who will be presenting their insights into how electrode manufacturing is shaping the future of biosensing technology. After a break, we will conclude the evening with a networking session, giving you the opportunity to reconnect with experts and innovators. Now, I hope you're all excited as I am. To officially kickstart this year's competition, it is my pleasure to welcome you to the stage, the chair of SensUs 2025, Alshra.
Hello, everyone. Once again, welcome to SensUs Kickoff 2025. It is the 10th edition this year, which means 10 years of teams working on socially impactful topics, and also 10 years of SensUs organization students working together to arrange the competition. However, SensUs is not only a competition. It is also a platform for you to learn so much more than the information we are given in our regular courses. Also, to meet new people and to expand your network. This year, we have a total of 19 teams, which means even more people for you to meet and to expand your network. As in previous years, we have 4 main award categories. The first one, Analytical Performance Award, will be given to those teams whose biosensors will have performed the most accurate measurements during the Eindhoven testing event. The next one, Innovation Award, is based on the novelty of the biosensing concepts that the team will implement and demonstrate in their biosensor. Third one, Translation Potential Award, will be given by jury to those teams who will have developed the best plan, the best strategy on how to transform their biosensor to real-life applications. And finally, the Public Inspiration Award, which will be given to those teams who will manage to inspire the public voters from all around the world that their idea is the best. And now, to have a quick peek into how the awards of the 10th edition of SensUs will look like. To arrange everything, we have a team of also students. Currently, we are with 16 people from 8 different majors at Eindhoven University of Technology, who will be trying to create an unforgettable experience for all of you. We hope that you will enjoy the upcoming months, and we are really looking forward to seeing everyone during the Innovation Days in August. To wrap up, I would like to suggest that we have a look at how the Innovation Days last year went during SensUs 2024.
Hello everyone. I wanted to welcome you to the final event of SensUs 2024.
Over the past years, the student teams have been developing biosensors for the continuous monitoring of acute kidney injury. Today, the teams are testing their biosensors for the continuous measurement of creatinine in interstitial skin fluid.
Hi there, I'm Tasneem from the University of Glasgow team, GLA Sense. This is my second year participating in SensUs, and to me, SensUs is all about teamwork, meeting people from different countries, and doing real-life engineering.
Today, the students are meeting the partners of SensUs. They have speed dates, workshops, visits and discussions.
It's all about having the team aligned with the customer in the right way to actually make the achievement together and feel that your customer is also part of that project.
So SensUs means to us getting in contact with enthusiastic students. It means to us telling the story of our company and showing them how our sensors work.
Today is the final day of the SensUs contest, and the teams are pitching in front of international juries and trying to win one of the awards.
What I like most about this SensUs competition and participating as a jury member is that this competition brings students from all over the world together, and it challenges them to participate in designing an innovative biosensor to tackle and solve a problem that is an existing problem in the world today.
What an exciting glimpse into last year's SensUs competition. And this year, we're taking it even further. Last year, our focus was on acute kidney injury, which is a critical area. But why are we continuing with this theme in 2025? To tell us more, I'm honored to welcome the founder of SensUs, Menno Prins.
Thank you very much, Mila. So let's talk about the theme of SensUs 2025. Over the period 2016 to 2023, SensUs focused on point-of-care testing. In point-of-care testing, a single sample is taken from a patient and it's measured on a single biosensor. Now this has a disadvantage because for every new sample, you need to take a new biosensor, which is not very efficient if a patient needs to be monitored and when a series of measurements needs to be performed. In that case, it's much better to have a continuous biosensor. And that's why in SensUs 2024, the theme switched from point-of-care testing to continuous biosensing. So in a continuous biosensor, a sequence of measurements is done in one and the same sensor without sensor replacement. And such biosensors are already available for patients with diabetes, so they can continuously monitor their glucose levels. Unfortunately, such sensors are not yet available for all diseases and for all molecules. For example, patients with kidney disease could benefit from continuous biosensing so they can take action before problematic symptoms occur. Now, in SensUs 24, we discovered something, which is that it was technically quite difficult to develop a continuous biosensor for kidney disease in just 1 year. And that's why SensUs decided to focus another year on kidney disease and to challenge the teams to achieve next levels in continuous creatinine sensing. So for the Analytical Performance Award, it's to show that your continuous biosensor functions truly continuously, without interruption. And to demonstrate that your continuous biosensor gives correct creatinine results. For the Innovation Award, to explain how a wearable creatinine sensor can be made with reliable calibration. And third, for the Translation Potential Award, to conduct customer interviews in order to collect solid data about the applications and to design a validation study for the sensor use that you have in mind. And these challenges are significant and they are difficult and they can have large impact. And the challenges are explained in the information package that you have received. So let me close this speech by wishing you and your team a lot of success. We look forward to your results, and if I can be of help, or if SensUs organization can be of help, please give us a call. Please send an email. So now, let's look at a video that describes the theme of SensUs 2025.
For the 10th edition of SensUs, we'll be looking back at our past editions and preparing for an exciting future. This year, the teams will develop continuous biosensors that are capable of monitoring creatinine, which is an indicator for kidney failure.
Just in case you don't know what kidney failure is, we'll try to explain it in just 20 seconds. In kidney failure, more than 85% of kidney function is lost, causing waste products like creatinine to accumulate in the body. Over 2 million patients are affected by it worldwide. Some symptoms include itchy and dry skin, nausea, fatigue, and muscle cramps.
Last year, the teams worked for the first time on continuous biosensing, showing that it is possible, but truly challenging. This year, they will take it to the next level and show to the world that biosensors can become truly continuous and suited for use by patients in the future.
Are you excited for the frontiers of biosensing technology being pushed? Then join us for SensUs 2025.
Yep. Thank you, Menno, for your presentation. And now that we know this year's theme, our first spotlight tonight is on Ardy van den Berg from PalmSense. This year's partner, PalmSense, is committed to making electrochemistry easier, more portable, and novice for advanced researchers. Today, Ardy will share valuable insights on how to accelerate your way out of the lab into real-world applications. Please welcome Ardy van den Berg joining us online.
Wait. Can you unmute?
Sorry for that. So my name is Ardy. I will give you a short introduction to what PalmSense is all about. I'm a business developer within PalmSense for over 5 years now. And before I start, it's very good to know that there are different ways to solve the task that you have at hand as a team of students. So you can, for example, detect the biomarker in an optical way, or you can use, for example, electrochemistry. And especially for the team who's using electrochemistry, this will be interesting. So I'll tell a bit more about when we were founded, so more than 20 years ago already, in 2002. We're based in Houten, small city close to Amsterdam, like half an hour drive. We're not a super big company. We have 35 employees, but we do sell all our instruments all around the world. So last year we shipped to over 100 countries. So almost any lab in the world will now have a PalmSense instrument, which is very nice. And we basically do two important things. The first is we enable research in the field of electrochemistry. So you have to imagine like a single channel or a multi-channel instrument with or without a battery that can be used, for example, in a glove box, as you can see in this image. So this allows you really to perform in a very portable way electrochemistry and perform all your research and develop biosensors. However, at some point your biosensor is done and you would like to move to the next level. This is where we help as well. So we enable biosensor applications. What you have to think of here, for example, on the left, you'll see an example from VPOT, a Finnish company, and they're using PalmSense instruments to detect painkillers in, for example, paracetamol in your blood, and if there's an overdoses. And it's very important because it could be lethal if you have too much of it. But you can see as well other applications like wearable applications and other continuous applications, continuous measurements. And this is what we really try to help with. So PalmSense delivers the hardware, and then you can focus on the sensor itself and the software. And at PalmSense, we have really multiple R&D disciplines under one roof. So we have electronics designers, mechanical designers, but also, of course, software and cloud development. At the right bottom, you see here a few photos of how it looks here. So where you can see our production facilities and our warehouse. And we're really innovation driven, right? So we take the latest trends and we implement that into our potential stats. But we also would like to help you as students and as student teams to accelerate you out of the lab. So most of the teams already know what you see here. This is the small potential stat development kit, the Amstel Pico development kit as we call it. And inside there is an Amstel Pico module, small potential stat module that can measure all your electrochemistry that you need to. And some of you might also know this smartphone potential stat. It's very small. And we've taken the chip that's in there and we've put it into a wearable device, which allows for continuous measurement on your skin, for example. And this will be very useful for the next thing. And this will also be my last slide of what we have to offer for the SensUs team and the SensUs students. We have on the left side an Amstel Pico development kit that we can offer for a large amount of discount. This will be very useful for in your lab. That's option 1. And option 2 will be a Sensus Wearable Development Kit, which allows you to use it actually in the lab, but also outside the lab on your skin. It comes with some example patches, and you can basically create your own patch connected to this device, and then you have a small wearable potentiostat with your own patch that allows you to measure, in this case, the creatinine on your skin. And that's something you can get it from us at a promotional price. And also, if you have any questions, for example, for us, if you'd like to have some advice, or maybe would like to develop your own potentiostat for some reason, you can always contact us for some info. You can do that via info@palmsense.com, and then we will help you as much as possible. All right, thank you very much.
From technology to its real-world impact. To truly innovate in biosensing, we need to understand the experiences of those at the heart of healthcare: patients, doctors, and researchers. Let's take a moment to welcome our esteemed panelists for this session. Valérie van der Vlier, who is a patient with the kidney condition and contributed to the Sensus competition last year as well. Stijn Konings, an internist nephrologist at Catharina Hospital. And 2 PhD students, Maud Linson and Livio Oliveira. Yeah. A very good evening to each of you, and thank you for being here with us. Let's begin with the questions. Valérie, you are a patient with a kidney condition. Could you please share your experiences with this matter, what troubles you encountered, and how Sensus participants might be able to help people like you?
Yeah, of course. I am an ADPKD patient, which means I have a large number of cysts growing in both of my kidneys. And by now I'm also a transplant patient because I got a kidney transplant about 2.5 years ago. With ADPKD itself, my first diagnosis was a good 20 years before I actually needed dialysis. So in that period, the whole big period, continuous biosensing wouldn't have really done much, but when I actually got towards the point of almost dialysis, or just after my transplant, it would have been wonderful because I was at the hospital 3 times a week at least to get blood draws and to get checkups and all those kind of things. And if you could then have a patch on your arm and just your doctor can just see what's going on and give you a call and say, okay, let's up the dose. Or when I just started dialysis, we really had to tweak the amounts of fluids and how much, which ones to use and everything. I did at-home dialysis. If my doctor would have just been able to call me and say, do this instead of me going to the hospital, getting blood drawn, waiting for an hour before the results were in and everything, that would have really saved me a lot of time and made my life easier. And if I look at what I think it would really be great for, it would be for like a friend of mine who really had acute kidney injury. She, from one day to the next, got really, really sick. And fortunately, everyone was on it immediately and figured it out quickly and put her on dialysis fairly quickly. But if in that time she would have been able to wear a continuous biosensor, that would have been wonderful for her because she was just freaking out the whole time. Is this working? Am I dying? It would give someone like her a lot of peace of mind, I think.
Yeah, thank you, Valérie, for sharing. And that is hopefully something that we will achieve someday with the Sensus competition. Stijn, as an internist nephrologist at the hospital, you have probably seen patients like Valérie. Could you please maybe share how a typical process of diagnosing and treatment looks like, from the doctor's perspective, of course, and how potentially different biosensors could impact that?
Well, thank you for this question. Just as you said, it's very important to have all the data, and the patients have to come to the hospital to the blood test and all those kind of things. But if this would have been possible with remote monitoring, then the patients don't have to come that often to the hospital, because when they have a chronic state of chronic renal failure, then it's really important that all the parameters are very stable. And normally we check this every 6 weeks or even more often if this is necessary. And then it would be very wonderful if we could monitor this on distance. And even when the patient is not feeling very well, as in the example of your friend, if you could do a test at home and we could see immediately what was going on, this would be wonderful. And this now patients are doing a lot of traveling, but this can be very easy by monitoring at a distance. So that would be wonderful.
Yeah, definitely, I agree. I think it would really make the process much easier, of course.
Yes, and also another point is, and I think you can remember this, a very important point is when you have chronic renal failure, when is the start to start renal replacement therapy or do a transplantation? And this timing is very difficult because sometimes the figures change very quickly, and then it is very nice that somebody can call, I'm not feeling very well, can we check the blood, and if it can be done by remote, even in the weekend, this would be wonderful. I think this would be very helpful for you in your situations.
Absolutely. I had actually, my creatinine was suddenly rising very, very fast, and I got my PD catheter put in, and then I stabilized, and I ended up, so for me, it was a little easier because I happened to already have a PD catheter, and then I didn't need it for 11 months.
Okay.
And when I suddenly started feeling bad, I called my doctor and said, I'm feeling this and this, what you said I had to watch out for, and I went into the lab, got my blood drawn, and I could start the next day.
Yeah.
And that was great because I happened to already have that catheter because I stabilized. But most people don't have that. And in those cases, that'd be wonderful to just be able to monitor and I knew exactly what the symptoms were to watch out for, but not everyone knows that, and not everyone's a geek like me who likes to know all the details about their kidney disease. And if you can just call the doctor and say, hey, can you check if I'm okay, instead of you having to say, like, I think I'm not okay, that would be great for patients, I think.
And even in the situation after renal transplantation, it is also very nice to do a blood check when you're not feeling very well, that you know
absolutely.
that the renal function is stable, because it's very important that they want to keep their kidney functioning, of course. So this would be wonderful.
Yeah, the amount of blood draws I got just after the transplant, just the first day when I was still in recovery, if that could have been done with a patch on my arm instead of every time stopping the IV for 10 minutes and then drawing blood and that would have been that would have been magnificent. That would have been really, really great.
I can imagine that would be wonderful. However.
Now, Maud and Livio, biosensors like those in the SensUs competition aim to address real-world medical challenges. From your perspective, what are some of the biggest scientific or engineering challenges in designing reliable biosensors for healthcare? Anyway.
I think one of the biggest challenge to design a sensor is to have good controls. So to actually know that you are measuring correctly and also continue to stay measuring correctly. Because if you are going to deviate with your measurement, this will also impact the results that will eventually go to the patients. And also, to have good, reliable data that is statistically significant.
I think in terms of engineering issues, I would say how to make those biosensors small, because at the university we research using large equipment like microscopes or in my case microscope, because I'm using optics. But there are also other fundamental challenges in the sense of creating new recognition elements, I think, for making biosensor continuous. So this is a more scientific challenge.
Thank you.
And I would say that the miniaturization would be the one engineering challenge.
Yeah, thank you for sharing that. And Maud, why did you decide to use biosensing by particle motion rather than any other type of biosensing in your research?
So I'm in a research group of nanoprints, and that's mainly the technique that we are using. And what I really like about this technique, that it's really single molecule. So for some of our sensors, we can really look at the particles and really see the single molecule detection. And I think that's, with our technique, that's one of the only techniques that actually can look at single molecules.
Yeah, that's very interesting. Now, Valerie and Stijn, as potential users of such technologies, do you also think that there are most important factors, or is there something else that you consider more important that should be incorporated?
From a patient point of view, I think ease of use. It has to be easy. I mean, you need to not be working on a PhD to be able to put the thing on and read it out, even though the results will go to your doctor too, hopefully, because I think that would be wise. It'd be nice to be able to see it yourself too, so that you get kind of a grip on it. For instance, if I think of myself, if you happen to have not had enough to drink one day and your creatinine spikes, it's not so horrible. If it's if you've been doing the exact same thing as always and it spikes, then you want to talk to your doctor. And it'd be nice to be able to see those kind of things and have it really be easy for the patient to work with it. Maybe an app on your phone or on your computer, something that's really easy besides the possibly more technical stuff that would be sent to a doctor in their system.
Yeah, that's true, I think. And I think the data should be reliable, so that's, that we can trust them, of course. And maybe we should also use some kind of AI that do an interpretation, because when it is only a little spike when you're not drinking, then the AI would say, well, don't worry, so that there's some kind of interpretation, because if all the data are coming to the doctor directly, then there will be a lot of data and they also have to be analyzed. So I think it's good when there's some kind of interface that filters, when there is really a sign that's really giving an alarm, that it points out a little change is not, but that the patient is always possible to do a check and see what the result is. I think that would be very nice.
Of course, but do you think it is wise to make use of AI at this stage, or do you think this is something that would potentially come later on? Because I don't know how reliable it is to fully trust the AI algorithms for this.
I think that will become later on.
Later on, yeah.
And now already we do in our hospital a lot of remote monitoring for cardiac disease and for COPD. And then we have a monitoring unit when nurses are looking all day for the data. And when they think that the data are alarming, they make a call with the doctor, of course. So there should be somebody who is interpreting all these data.
Okay.
And we have a remote center in the Catharina Hospital for heart failure and for and we also now going to start this for hypertension and also for chronic renal failure.
That's awesome.
Yeah.
Which will be very nice.
Yeah. Oh, absolutely.
And there are always nurses you can call if you have a problem, and they can look at the data. And when they say, well, these data are really alarming, then they get checked with the doctor, of course.
Exactly. That's perfect. That's from a patient. I think that's really nice.
Yes.
Yeah, thank you for sharing that. Now, Maud and Livio, your research focuses on continuous biosensing, which, as we have seen, could significantly change how we monitor health conditions. Maud, your work on IL-6 detection has implications for inflammation, sepsis, and also organ transplantation, while Livio, your work with plasmonic materials aims to enhance biosensor performance, if I understand correctly. What are some potential real-world applications for your technologies that you see, like beyond the lab, and how do you see them benefiting patients in the future?
Yeah, for me, I'm already quite closely working with doctors that perform transplantations. So we are really looking on how our technique can be used for beneficial in determine if an organ is still viable for transplantation or not, because there's currently a large shortage of donor organs. And we are looking into better systems to get the organs, to revive them and make them better for transplantation, and also to give a better assessment if the organs are viable for transplantation. And furthermore, the research can also help in looking at cell-based systems that might eventually help to go from animal testing to more cell-based system testing for instance, pharmaceutical industry.
It's very interesting.
Yeah, so for me, I'm using very, very small materials. So I'm using, you said plasmonics, so just to put that in context, those are very, very small particles that are invisible to the eye. So I'm using a microscope to observe them. So what I aim in my research is to push the sensitivity of the biosensor. So here we're talking about kidney disease, which has one specific marker, which is creatinine. But we also want to generalize to other types of targets. And some of them, they are found in the body in very, very low concentrations. So my research is to push the limit of detection of targets. Yeah, and also I'm now conducting also research on how to make those biosensors, how you can use them in complex fluids. So in the lab, we usually start with buffers, which are solutions that they just mimic a bit the conditions of the body fluids, but we also need to test them in real life settings. So testing also in human serum and other types of fluids.
Yeah, well, thank you very much for sharing about your research. And of course, hopefully in the future, we will achieve more applications of that, and not only for, of course, using the biomarker of creatinine for this. Now, Stijn, as a doctor, you're in some sense between patients and engineers. From your experience, what makes these collaborations between engineers, clinicians, and patients effective or maybe challenging?
Well, I think it's very important that there's good communication between all the different persons for the use in the clinic. And when you introduce new techniques in the clinic, there's a good communication. But normally, with a good interface, this is, in practice, this always goes very good in our hospital. So when there's a new technique, we have our technicians, we have the doctors, the patients, we talk together how we want to use them and interpretation. So I think short lines of communications by using these new techniques and then do some kind of validation, and then you can use it in the clinic.
Yep, well, thank you for sharing that. Now, this is a question for all of you. If you were a student in the SensUs competition today, what aspect of biosensor design would you focus on, and what impact do you think it would aim to make? Like if you were to put yourself in the position of a student working on this biosensor right now.
Accuracy and wearability, I think, would probably for me be the most useful. Since the idea is with the innovation that that it should be able to indeed be used continuously, so it needs to be wearable, easily wearable. No patient's going to run around with a giant box or something. So wearability, but definitely accuracy. That's even more important.
Yes, I think you can think about the glucose monitoring, which has become very popular. The Freestyle Libre, maybe you know this system. That's a system that's very easy to use by patients. It's very easy to communicate. And when the patient comes to the doctor, the patient can show all the data, so that is just easy to handle, and that is reliable. That's very good,
Okay.
I think.
Yeah.
And this, maybe you know this system. There's a Freestyle Libre. It is used for patients with diabetes. So they only have to go with the iPhone on the sensor, and you get all the data, you get the interpretation, and you can also see in the long run what was happening, what was the condition of the patient, so that it is easy to show to your doctor when you go to the consultation, and then you can take your own data. You don't have to go to the lab. You just take your iPhone and you show what was going on with you. And this can be combined with other data like weight or blood pressure. Those are the most important things we look at. So if it can all be collected in one system, it would be very nice, I think.
Yes, yeah, definitely. I agree with that. It would definitely increase the usability and also just make it more convenient for patients and everyone else and doctors as well. Yeah, Maud and Livia, do you have to
Yeah,
add
I
to that
have
as well?
more of some tips to the teams. I would recommend them to have a diversity of people of different backgrounds that can focus on different parts of the biosensor. So since the recognition element, the transduction, as well as also talk to people, to patients, ask them what they need, ask a lot of questions, talk to professionals, because you can get a lot of insights with that. And in terms of as Maud also pointed, have good controls, run experiments, try to be accurate, try to be precise, and there's a lot of elements in the competition that can be very valuable for you, and most importantly, also have fun in the competition. I think it's important.
Of course.
Yeah, I think one important aspect to focus on is to have also good reproducibility, so that if you have a good result once, doesn't mean that you also will get a good result the second time. So also try to make a sensor that is that you can make again.
Yeah, thank you very much for sharing the tips and tops for the teams.
Oh, I have another one that I just thought of. I was here last year too, and there were a couple of teams who already talked about the possibility of sensing multiple things, like for instance potassium, and if that would definitely, I think Stijn would agree with me that it's not just the creatinine that makes whether you have to start on some meds and everything. So yeah, if you do have the possibility to do something like that, or to at least get that idea going, even if you don't have that part working yet, that would definitely be a plus in my book.
Yeah, that's right. We need more than only the creatinine. So what's possible to give very easily, like the potassium is very important, the phosphate, the total CO3, the acid base content, that would be very nice. So to give more data, that's right. Because there are more factors than only the creatinine, that's right.
Yes, of course, of course. But I also think it's difficult to measure some of the biomarkers, which is why I think the teams are focusing mainly on creatinine at the moment.
Yeah, it is good to start with, I think. But
yes.
In the future, I think there will be more possible, because it's an evolving
of course.
type of research you're doing, I guess. So
yes, yeah.
Maybe in the future there might be more possible, I hope.
Well, this wasn't possible 10 years ago. So.
No, that's right. And, so the things are changing, but I think for the patients it would be very nice if there would be more possible, that they can get more freedom, of course.
Of course.
That's important for
you. Absolutely.
Yeah, hopefully moving forwards in the future, that is something that we'll be able to achieve. Well, thank you for ending on this inspirational message to end the panel talk on, and inspiring the teams, of course, in this competition. Thank you all very much for answering the questions and for sharing your insights and invaluable knowledge. It has been a pleasure, of course, having you here, and we have some gifts for you to thank you. Yeah, these are some gifts on behalf of SensUs.
Oh, okay, thank you.
Thank you.
Yes, go.
Thank you.
Yes. Now, as we've wrapped up the panel discussion, it's time for our second company presentation by Katalin Ramsamy from LinkSense. LinkSense is a global leader for more than 40 years in the electronics industry, playing a key role in advancing biosensor technology. Katalin will share insights into how innovations in electrode manufacturing are driving the development of high-volume, cost-effective point-of-care biosensors. Please welcome Katalin, joining us online from France.
Hello everyone, do you hear me? I think yes. So yeah. So if we can make the correct diagnosis, the healing can begin. If we can't, both our personal health and our economy are down. And this is an advice that we have from from Andrew Well about an advice to have a good device for biosensors. During your competition, your challenge will be to develop a kidney solution for diagnostics. Whatever the shape and whatever the version and the solution that you want to develop, your challenge will be to have something stable, something with a high quality, something reliable and reproducible. And for that, during your journey, your challenge will be to find the good biosensor transducer, and there is a lot of solutions onto the market. Among these solutions, you have screen printing electrodes. It is a well-known solution. A lot of people buy this kind of electrodes for experiments, but unfortunately, with this kind of electrodes, you have a problem on the reproducibility. You have also another options. You have spot-read electrodes, pure metals, good precisions, but the problem is the cost because it is an expensive solutions. You have also PCB electrodes. It's a good compromise because you have the quality of the spot ring electrodes because the quality is good, the cost is okay, and the stability also is okay. With LinkSense, we develop flexible PCB, and the advantage of our electrodes is the usage because you don't need to clean this electrode before the usage. Our reproducibility is good, and the price also is competitive in comparison with a spot ring electrode and with also screen printing electrodes. And we are able to develop a solution with complex structuration. And I can give you some information on our process. All our process is in row-to-row. So that means that the electrode will be manufactured on a reel. And with this option, you will have this opportunity to customize your electrodes. So if you want to develop an electrode with a different shape or different structuration, thanks to our technology, thanks to our process, you will have the possibility to do it. And you will have also the option to add additional components. So for example, you can add a chip or SPO2 sensor or whatever kind of sensor is that you have in mind. We have the facility to propose this service. And if you want to add on your sensors microfluidic systems or laser disconnecting or things like this, we have also these solutions. About the constructions, we can develop with you different constructions. So we have single side structuration. We can have more complex structuration like diverse sites. And we have also the possibility to do well systems. So that means that your electrode will be inside a hole. As I said before, we can do different shape and we have a large quantity of possibility on substrate and on metal finishing. So that means that in function of your need, we will be able to customize the electrodes with you. And we have an example on the market. If you well know Siemens Healthineers, we developed with this company the biosensors, and it is a biosensor for blood gas testing. And we are producing these products in millions of pieces. So with LinkSense, you will be able to produce your biosensor, but you will be able also to integrate your biosensor inside patch. For example, if you want to develop something for continuous monitoring, we have the facility for that. And we can also work with you to develop solution on track and trace. So that means if you want to develop something with your biosensor, with a patch and with RFID connections, we will be able to provide also these solutions. So if you are interested to develop your biosensors with us, we have the possibility to send you free electrodes to test it. Our electrode is also available on PalmSense website, and don't hesitate to contact me if you want to share with me your project for this competition. Have a good day.
For diving deep into the electrode technology and sharing your expertise with us. With this, we conclude the official part of the event and encourage everyone to actively participate in the networking session that is about to take place. Everyone should have received the schedule on Canvas, and there are team invite links as well to the channels. We hope to see everyone at the networking moment and have a fantastic evening. Good luck with the competition.